All right, hello everyone, and welcome back to our ML for Protein Engineering seminar. We had a little bit of a hiatus over the summer, but I'm really excited for today's speaker, Yo Akiyama, who's going to be telling us about MSA Pairformer. He's a grad student in Sergei Ovchinnikov's group at MIT, and I'll let him introduce himself a little bit more and get right into the talk. But if you have any questions, you can leave them in the chat. Or you can just type the letter Q and then we'll let you unmute. I think Yo is going to pause at like different points in the talk to take questions, so you can feel free to leave them in the chat while we go. Otherwise, we'll take most of the questions at the end. But yeah, take it away.
Yeah, thanks so much, Jason. And thanks for inviting me to give this talk because I've been tuning into these for a few years now and I've learned a lot. So it's a big honor for me to sort of give one of these talks myself. But yeah, as Jason said, I'll be talking about MSA Pairformer. This is our new model, this new protein language model we've been working on, and how maybe we're challenging sort of the paradigm of scaling laws, and at the same time sort of expanding the scope of what these protein language models can do. So as you said, I can stop sort of at certain points in the talk to take questions. I'll let you guys know when those times come up. Can y'all see everything all right, or is the meeting control sort of blocking some stuff? Or
That's good.
maybe I can. Okay, sounds good. Cool. So just a brief outline here. I'll just give like a very short overview of protein language modeling for anyone who's not super familiar with them, and sort of how people are trying to make them better. And then what problems sort of those approaches run into. And then we'll talk about how our approach, MSA Pairformer, sort of addresses some of those challenges. We'll walk through the architecture and then some results. And then we'll also go into some sort of interpretability studies we've done with the model. So just very briefly. So like protein language modeling is super simple, right? You just train a model to predict missing the amino acids at these like randomly masked residues of a protein sequence. So, and then you repeat this across the protein universe and train the model to just predict these sort of hidden amino acids. And through this process, right, it sort of learns to maybe capture evolutionary constraints that are sort of applied to these residues. And in doing so, we hope that the model is sort of learning like either maybe grammar protein language model or proteins, or maybe sort of storing evolutionary information in the model. And then we can sort of generate embeddings for proteins and use them for sort of prediction paths downstream. So some things we can do with them is like see if the model is learning sort of structure just from this sort of masking, unmasking process. And another thing we can do is sort of use these embeddings that the model produces to predict certain properties of protein sequences. And so that's like the very basic sort of simple magic of protein language models. And so now we're just trying to sort of improve them, sort of expand them to different problems, and sort of hope that they are able to capture different properties of proteins across the protein universe. And so how do people do that these days? Well, the focus is mostly on scaling, right? So as people have seen in sort of these like language model studies, like the bigger you make the model and the more data you feed it, the better it'll get. And so people have taken that to heart with protein language modeling as well. And so you see a lot of studies that sort of scale essentially the same architecture, but just make them a lot bigger, train them on more data. And of course, you find that it's perform better on like the actual training task, which is just like predicting masked amino acids. And with like the ESM2 family of models, they've seen that making the model bigger actually improves sort of this like unsupervised contact prediction. So maybe the model is like these larger models are able to capture sort of protein structure better from just the sequence alone. And there's also a lot of work on expanding sort of the data set that these models are trained on. So there's like the Progen 3 family of models was trained on this like massive new data set. You have like people focusing on sort of synthetic protein sequence generation. And this sort of new paper here is just like expanding these data sets even further to sort of capture sort of more viral proteins as well. So these are like the major paradigms of improving protein language modeling right now. And so sort of the idea for doing this is very straightforward, right? The model seems to work better at a lot of tasks when you scale them up. And so like, why might this be the case? Well, there's been sort of interpretability studies, one from our lab here, that sort of showed that protein language models are essentially storing evolutionary information within the parameters of the model. So I guess the larger your model is, the maybe the higher resolution information it can store within the parameters. And the more data you train it on, of course, the more sort of sequences or the more information you're able to store in the model. So this sort of explains why maybe the models are doing better at this like masked amino acid prediction task and potentially this sort of contact prediction task as well. And another sort of thing we've seen here with the Progen 3 families is Progen 3 is more of like a generative model because it works autoaggressively, and they found that the larger you make these models, or the more of the natural sequence space it can cover when it generates protein. So it's pretty clear that these models are essentially just storing evolutionary information within the parameters throughout training. But why might this be a problem, right? Well, for one, right, scaling is like very expensive. When we make models sort of like billions or even sometimes trillions of parameters, they cost a lot to train, and they also cost a lot to just use, right? And so that's on one hand, it's super expensive. On the other, if we're just storing this like evolutionary information within parameters, we might ask, like, is this process sort of cursed, right? Like the amount of available sequences is growing very rapidly, right? And so the more data we sort of sequence, maybe the larger we have to make the models, and then this process becomes more and more expensive and sort of unsustainable. So how can we sort of move beyond this? And another thing is people have shown that scaling doesn't always help for all tasks, right? So the Pascal and Debbie Marx's lab has sort of seen that if you scale these protein language models, after a certain point, the models tend to do actually worse on zero-shot variant effect prediction. And there's been some work on sort of explaining why this happens, but it's sort of clear that performance doesn't sort of monotonically increase across all sort of prediction paths. So we want to be able to advance protein language modeling, and that means we want them all to get better at all tasks and not sacrifice sort of ones for others. And so one alternative to scaling is that so if you single sequence protein language models are essentially just storing evolutionary information in their parameters, we can just provide that information at inference time. So this would be more of a retrieval-based approach where we essentially just query a protein sequence of interest to this massive protein database and produce this multiple sequence alignment. And this alignment essentially gives us sort of the evolutionary context for a given protein by essentially just retrieving sequences across many species across the tree of life for a given protein. And we can sort of use this information at inference rather than training a model to store it in the parameters. And so this allows us to have much smaller protein language models that potentially perform just as well. And another benefit here is that since the model is just training to extract this information from a set of sequences, we can always add new sequences to multiple sequence alignments at inference time that we never trained on. And so this allows the model to sort of be more adaptable to this newly available sequence data that we've sort of sequenced after training the model. Another thing that's great about multiple sequence alignment models is that we can always sort of subsample multiple sequence alignments or sort of cluster sequences within an MSA. And so this allows us to have sort of more flexibility with sort of the context, the evolutionary context that the model is looking at for a given protein sequence. So just a graphical overview here, of course, like single sequence models like ESM and Progen 3, they take a single protein, mask some positions, or in the case of Progen 3, they mask the next position, and then they're just training to predict the missing amino acids in that one sequence. And the difference with an MSA-based model is we give it an MSA, and then we mask some random residues within the MSA, and then the model just trains to recover those masked amino acids. So it's very similar, but we're just using multiple sequence alignments rather than single sequences. Of course, this approach is not super revolutionary or anything, right? We've seen this work very well in structured prediction models like AlphaFold2 and AlphaFold3, Rosetta Fold, all those other supervised structured prediction models. And with those models, it's very clear that the multiple sequence alignment is actually very crucial for these models to predict the final structure. And then the thought is that the model is extracting sort of these pairwise relationships between positions of the protein through sort of evolutionary information. to predict the final structure. And so if we want to make a multiple sequence alignment model for self-supervised protein language modeling, there's sort of some things we need to address. And I guess like the main limitation we find with current multiple sequence alignment methods is they have this assumption that all of the sequences in a multiple sequence alignment are sort of equally relevant to the query sequence, so to the sequence we actually care about. And so they do this like averaging of signals across all the sequences in the MSA. And so with MSA Transformer, which is sort of like the state of the art sort of self-supervised learning MSA-based model, they do this tied row attention where they make an attention matrix for all of these sequences, and then they average them together to make one attention matrix for the entire of the MSA. With outfold 2 and outfold 3, they do this outer product mean, where they, when they compute the interactions between residues of the protein, they average those across all the sequences in the MSA. And so there's like this explicit averaging that these models do that maybe is not sort of conducive to what we want with a protein language model. And the reason for that is that in these large protein families, you might have subfamilies of sequences that are more evolutionarily related than others. And these subfamilies might contain sort of distinct functional and structural properties. So they might sort of make different amino residue-residue contacts, or they might make different heteroligomeric or homoligomeric contacts with other proteins. So we want our model to be able to capture these differences within a large protein family. And so this averaging mechanism probably isn't going to work very well for us. Another thing people have shown, so this is like Hannah when she was working Brandeis, but she found that sort of clusters of sequence within an MSA might encode for sort of different conformations of a single protein. So we want our model to sort of be able to do this, maybe clustering in this unsupervised way, and learn this end to end with just fast language model training. So we'll talk about how we're going to do that. But yeah, so to motivate a new protein language model, right, we want one that's sort of memory and parameter efficient because we want people to be able to use it. And we want it to be able to adapt to this rapidly growing protein sequence databases so that we can sort of leverage new information that's often very helpful for predicting stuff like structure or function and stuff like this. We also want the model to be able to capture subfamily specific evolutionary signals like we just talked about. And lastly, we maybe want to expand beyond the current capabilities of protein language models. We'll discuss this a bit later when we start talking about protein-protein interactions. So we first introduced MSA Pairformer here. And so this is our basic architecture. We're essentially just building on outfold 3's MSA module and using it for self-supervised protein language modeling. So rather than training it on maybe structure prediction like they do with outfold 3, we were asking, hey, does this work for self-supervised mass language modeling? And so the architecture is actually pretty simple. It does this bi-directional refinement between sort of the sequence representation that you would sort of is more analogous to what you would see in like ESM2 or like MSA Transformer. But we also we update this like pair representation that allows the model to sort of throughout the model sort of refine the relationship between positions of the protein. And to do that, we're going to use these like triangle updates. And we'll talk about more about what those might be doing in the model later. But for now, we can think about this as maybe refining the relationship between residues of the protein and using that information to then update sort of the representation of the sequence. And so we do this sort of bi-directional refinement of both. And we think that this is actually probably going to be very helpful for even this protein language modeling task. But importantly here, we only backpropagate the loss on this fast language modeling objective. So this is not supervised on structure at all. If you notice sort of this middle part here, it says the outer product mean, and that we just talked about how maybe that is not the most appropriate thing to do for an MSA-based model. So how might we address this? So rather than computing just a basic average over all the sequences in the MSA, we were wondering if we could have the model sort of infer the evolutionary relevance of sequences to the query sequence. So the query is the one that we're most interested in. So can we use something like attention to have the model compute this relevance term? And so what we do is we just take the query sequence, and per column we compute an attention score using dot products. And then we average that across all the positions to make like a per sequence relevance score. And then we use those to compute this weighted outer product. So this is very similar to the outer product mean, but rather than doing sort of this uniform averaging, we take this weighted average where the weights are determined by this attention score. And then this updates the pair representation. So in the end, we have this query biased outer product where we replace the outer product mean before, and perhaps this will allow us to capture these subfamily specific signals within a multiple sequence alignment. So I can take questions right now if there's any sort of clarification questions that need to be addressed.
Yeah, you can
Cool.
leave them in the chat or raise your hand. I also am allowing unmuting now if you want to directly ask. That doesn't look like there's anything.
Cool. Okay. Well, if not, we can move on to some results. Cool. So the first thing we want to see is, does the model capture structure from the sequence? And of course, like we have these like supervised structure prediction models, so we don't really care that the model is able to predict structure or anything, right? But what we want is to know that the embeddings of the model are capturing or at least considering the structural properties of the protein, right? Because we know that structure is very important for even the function and stability, whatnot of proteins. So we want these embeddings to consider the structural relationships between residues. And so the way we can do this is say, okay, from this mass language modeling objective, does the model sort of understand structure? Can it extract residue-residue contacts within a protein? And so we use Cas15 to sort of evaluate this. And what we can see here is just like how models like ESM2 improve in this unsupervised structure prediction task as the models get bigger. And here we also have MSA Transformer. So the x-axis here is just how big is the model. The y-axis is sort of how well does it do at contact prediction. And so what we find is that MSA Pairformer, even though it's roughly over 100 times smaller than ESM2, is that the MSA Pairformer actually performs a lot better at this long range unsupervised contact prediction. So this was a pretty exciting result for us. And if we look at some specific examples, we can see that in examples where ESM2, and if we use just this like traditional like statistical based method, we can't accurately predict contacts between residues. But what we found with MSA Transformer, and here actually MSA Transformer does all right, and then MSA Pairformer does quite a bit better. So we find that the models seem to be leveraging the information in the MSAs a lot better than single sequence models. And one thing, just to go back with to what I mentioned before, is that people have noticed that sort of if you scale single sequence models, at a certain point, the models get worse at this at zero-shot variant effect prediction. And so we wanted to see if that was the case for MSA Pairformer as well. So we used protein gyms, sort of roughly 200 deep mutational scan experiments to see if the model can predict the effects of mutations on properties like activity, fitness, or stability, whatnot. And it's nice to see that we don't sacrifice sort of a zero-shot variant effect prediction for this sort of this increase in performance and contact accuracy. So this is like not like a huge difference, of course, but we want to make sure that we weren't sacrificing sort of variant effect prediction over contact accuracy. So going to the subfamily specific signals we talked about before, we can sort of use this response regulator superfamily as a case study for this. So there's this very large protein family of response regulators, and for three of the subfamilies in the response regulator family, we have crystal structures. And what we notice is that the homoligomeric interface. So this protein forms a homodimer, and we find that these three subfamilies actually bind at three different interfaces. And so if the model is able to sort of capture this like subfamily specific signal well, we should see that the model sort of predicts contacts for these very specific and distinct interfaces. And so what we do is we take a single MSA, and that MSA contains sequences from all three subfamilies, and we're going to predict contacts just by swapping the query sequence with a member of each of the subfamilies here. And so if the model is able to sort of extract the subfamily specific signal well, when we swap the query sequence to say this subfamily here, we should hope that the contacts for this interface are more emphasized. And so if we just do like uniform weighting, if we don't do any sort of weighted averaging for this outer product, we see that we sort of drown out signal for the homoligomeric interface, and we sort of get this mix of just a few contacts from these two subfamilies. Now, if we use query biased outer product and we swap the sequences, we swap the query sequence for members of these subfamilies, we find that in this OMPR case, we see the strong increase in this homoligomeric interface here. And for this GRE subfamily, we see this increase in the subfamily specific contacts here as well. And so this was cool to see. It sort of shows that this query biasing is allowing the model to sort of extract the subfamily specific structural properties. Another thing we can do here is sort of look at the distribution of the sort of the inferred weights that are used in the query bias outer product. And what we find is that when the query sequence is some member of some subfamily, we find that the subfamily members are often significantly upweighted. And so here, where it's orange, it means that the sequences belong to the same subfamily as the query sequence. And what we see across the examples is that the subfamily members are significantly enriched in their sequence weighting. So and we also don't see this like perfect correlation with Hamming distance. So I guess like sequence identity to the query. So this is also good to see because we know that some subfamily members are going to be quite distant. Like this example up here with GRE is like less than 10% sequence identity, but we're still able to sort of upweight that sequence here. And so the model seems to be using something beyond just sequence identity to capture the subfamily specific information. Cool. And so one thing we can sort of look into in terms of how can we expand the scope of protein language modeling is look at, well, does MSA Pairformer seem to be able to capture the effects of or capture the relationship between proteins? So this is sort of predicting protein-protein interactions without actually training on any sort of dimers or any protein-protein interactions. And of course, like, why do we care? Beyond just like, predicting protein-protein interactions, we want to be able to sort of use these protein language models to engineer and predict properties of protein-protein interactions. And so here we ask if MSA Pairformer can capture these PPIs, and if it can do it at residue-residue resolution at the interface. And so to test this, we use 31 protein-protein interactions with crystal structures from E. coli. And what we find is that MSA Pairformer substantially improves over models like MSA Transformer and GLM2, which have shown sort of evidence before that maybe sometimes they can predict these protein-protein interactions. But we see these like very large gains with MSA Pairformer here. So it seems like MSA Pairformer is sort of strongly improving over these other models. Interestingly, you note here, and maybe not too unsurprisingly, right? You see ESM2 really struggles to do this because it's trained on these monomeric sequences, and it sort of never generalizes to sort of figure out the interactions between proteins. If we look at individual examples, we find that MSA Pairformer consistently outperforms MSA Transformer. And we find that MSA Transformer seems to sort of suppress interfacial contacts quite strongly. And if we look at a specific example here with this ABC transporter protein, we see that actually MSA Transformer doesn't capture the protein-protein interface at all. And so we'll talk about maybe why this might be happening later. But it is really interesting because it sort of seems like MSA Transformer isn't actually extracting co-evolutionary information all the time. We know that there's co-evolution here. If we look at the inverse covariance, we see this sort of signal pop up for the protein-protein interface. So it's interesting to see that maybe some models, even if they should be capturing or if they appear to be capturing protein-protein interactions, sometimes they might not be actually relying on co-evolution. So yeah, another thing we did here was sort of improve how pairing, how to pair MSAs. And so this is a really important thing because what I just mentioned here was that the model seemed to rely on co-evolutionary information, right? And so we need to properly pair the MSA such that the co-evolutionary information is sort of stored in these sequences. And this is actually quite a hard problem because you often have these sort of duplication events in genomes that cause these protein-protein interactions to sort of duplicate and then evolve separately. And so you have this homolog or ortholog paralog problem where paralogous proteins don't actually interact with the protein you're interested in. And so we need some way of sort of filtering these out when we make our MSAs. And so this is where we sort of collaborated with Martin Steiniger's group to build something into MMSeq2. And so what we do here is we sort of leverage the operonic structure of prokaryotic genomes such that where the sort of interacting proteins are often proximally encoded in the genome so that they're sort of next to each other physically. And so what we do is we generate an MSA and then we can filter for pairs of sequences in close genomic proximity. And so when we do this, so if we don't do this, of course, we find that the paralogs sort of like pollute the signal in the MSA and sort of drown it out, and we don't predict any interaction between these two proteins. If we just maybe set a coverage or a sequence identity threshold for this MSA, we also don't recover any. But if we filter for proteins that are proximal in the genome, that we're able to properly recover these signals. And that's because the sequences in the MSA are now sort of cleaner and more representative of this actual sort of protein-protein interaction evolution. So, and interestingly, when we don't filter, it sort of shows that MSA Pairformer does hallucinate these contacts. And so that sort of opens opportunities for screening or potentially using MSA Pairformer to improve MSA pairing for maybe harder pairs where maybe like eukaryotic protein-protein interactions. Okay, so another thing we can look at for protein-protein interactions is since MSA Pairformer seems to be modeling these quite a bit better, we sort of hope that it's able to sort of model these and predict the effects of variance at PPI interfaces better. And so we use this sort of toxin-antitoxin example from bacteria, and it's very simple and it's easy to read out experimentally. And so we use this previous study from Mike Lobb's group at MIT, where they sort of take this toxin-antitoxin protein from E. coli, and they mutate the interface at 4 different residues at the interface. And so if there's no binding between these 2 proteins because the mutation sort of ablates interaction, the toxin is sort of on its own and it's able to sort of induce toxicity in the bacteria, and the bacteria dies. And so you get this sort of lower fitness readout when the mutations break this interface. And so we want to see if MSA Pairformer can better predict when a mutation will sort of break a protein-protein interaction. And so when we score just based on pseudo likelihood, we find that the sort of higher fitness binders, so we say they're higher fitness because the toxin and antitoxin sort of bind, so the toxin isn't released and the bacteria sort of is able to grow. And we find that these higher fitness binders have a higher pseudo likelihood score when scored with MSA Pairformer. And if we compare across models, we see that MSA Pairformer outperforms MSA Transformer, and the gap between these 2 models and ESM2 is actually quite large. And so to figure out if this sort of performance boost is from its ability to model protein-protein interactions, we did one experiment where we just score these sequences using only the toxin sequence. And when we do that, that's essentially just saying, oh, we're just modeling this toxin sequence independently of the antitoxin. And if we do that, the performance is very close to ESM2. So that suggests that this gain is sort of directly related to its ability to predict or model protein-protein interactions. And lastly, for results, we just checked how does MSA Pairformer sort of, how does its memory sort of grow with MSA depth and sequence length. And we find that just, of course, due to architectural decisions, MSA Pairformer is able to include a lot more sequences in the MSA, and this sort of will allow it to sort of use more evolutionary context, and maybe this will be very helpful for including information from really large protein families, or when sort of these protein data sets become, or databases become very large, we can continue to increase sort of the scale of our MSA as these data become available. So now is actually probably a good time to answer some questions if you have any. I'll just go into some
Yeah.
interpretability studies
I see
right
a
after.
lot of questions in the chat, so it
Oh,
might
gotcha.
be
I see them
better
now.
to just wait till the end because there's
Gotcha.
quite.
Yeah, let's wait until the end. That's a good idea. Okay, so I'll just jump into some interpretability studies, but I guess like something with protein language modeling is that it's hard to figure out what the models are doing. And so we're wondering if we could figure out sort of at least some components of MSA Pairformer and how they work. And we were wondering if sort of different architectures, so say MSA Pairformer versus MSA Transformer, learn different solutions to essentially the same problem. Um, and so to do this, we first focus on triangle operations. We thought that this is a good sort of maybe opportunity to look at what triangle operations are doing. And of course, these triangle operations, these multiplicative updates and triangle attention were introduced by ALPFOL 2. And they're also part of ALPFOL 3, of course. And this was sort of to build in this inductive bias that the model should sort of care about these geometric constraints between like triplets of residues when sort of predicting the structure of proteins. And of course, we use triangle updates in MSA Pairformer, and we're wondering what it might be doing in MSA Pairformer. And we sort of had this hypothesis that it might be disentangling direct and indirect correlations. And this would be pretty similar to what these like traditional statistic-based models do. So sort of like an inverse covariance matrix or direct coupling analysis. Of course, when you do something like mutual information, you only consider, I guess, like pairs of residues, and you're unable to sort of disentangle when two residues are just correlated because they share some contact with a third residue. And so we're wondering if if triangle updates are sort of allowing MSA Pairformer to remove these by considering triplets of residues. And so we did this ablation study where we replace this triangle updates with what we call pair updates. And with pair updates, we just allow the model to look at an individual residue and pairs of residues, but never triplets of residues. And this sort of forces MSA Pairformer to only look at double or pairs of residues. And it's sort of a harsher ablation than what people have done before, where they've done more like sort of axial attention, which maybe allows you to look at like two edges of a triangle. And so here we only allow it to look at one edge. And you might think that this is actually fairly similar to something like mutual information. So you have maybe your marginal distribution when you look at just your single residues, and then when you look at pairs of residues, you have joint distribution here. And so what we do is we replace triangle updates with our final model, and then we fine tune it with this pair updates to see what triangle updates might be doing in this like fully trained model. And then we also wanted to see if you could train pair updates to be just as good if you train it from scratch. And so we do these two separate experiments. And with these two, we also, to figure out what they might be doing, is we predict contacts for proteins using these two models. And then we check for whether the false positive pairs of contacts are enriched in pairs of residues that share a contacting residue. And so this will allow us to sort of see if indeed MSA or if triangle operations are sort of allowing the model to disentangle indirect and direct interactions. And what we find is that seems to be quite the case. So these false positive pairs, when you sort of replace triangle updates with pair updates, are often enriched in these examples where you have a third residue that is probably mediating that correlation. And we find that if you use triangle updates, sort of the false positives that you get from the pair updates are significantly subdued. And so this was sort of like an opportunity to sort of look at what triangle updates are doing with our model. And so we were really interested in sort of expanding this further and looking at sort of other components of the model as well. Another thing we can look at is, do MSA Transformer and MSA Pairformer behave differently? And something that was really interesting about the MSA Transformer paper was that they did these sort of MSA perturbations where they shuffled positions of the MSA. And so that's just asking, does the model care about sort of the positional relationship between residues? And then they also shuffle the amino acids within a given column. And so if you shuffle within a column, throughout all the columns, you should essentially remove co-evolutionary signal between the columns. And so, and what they find in MSA Transformer is that even if you shuffle the columns and get rid of co-evolution, the model is often able to sort of recover contacts. And we sort of argue that that's not a good thing. You sort of don't want the model to hallucinate context when the sequences that you provided are not going to fold into that structure. And so we want to check if this is also the case for MSA Pairformer. What we find is that MSA Pairformer does not recover these contacts. And so this sort of suggests that MSA Pairformer is sort of directly relying on coevolution or covariance between columns of an MSA. And we argue that that's a good thing, right? We don't want the model to sort of think that the sequences are going to fold into some structure when they're likely not going to. And so it seems like MSA Pairformer and MSA Transformer are learning actually quite different things, and they're predicting contacts using sort of a different mechanism, and perhaps that's architectural. And so a little open question here is that MSA Transformer is architecturally very similar to ESM2, and so there's a sort of transformer-based architecture sort of push MSA Transformer to operate more like a single sequence model. Yeah, so in summary, we sort of challenge the scaling law or this paradigm of making models larger to sort of hope that maybe they get these more sort of emerging capabilities. And so what we did was we trained a smaller model, and we're able to sort of improve on performance in these like sort of in these like gold standard evaluations, but we're also able to sort of expand the scope of protein language modeling to protein-protein interactions. And this is something we're mostly excited about here, because other models like ESM2 and even GLM2 don't sort of when you look at protein-protein interactions really get outperformed by even just like basic statistics methods. And we were also excited to sort of share this sequence, this MSA pairing tool that we sort of built into the MMSeq server. And so with this better protein-protein interaction modeling, we're hoping that we can maybe use protein language modeling to sort of engineer synthetic circuits, so like these two component systems within in vivo, and that'll sort of expand sort of the applications of protein language modeling. And lastly, we looked at sort of what the model might be doing, what certain components of the model might be doing, and we looked at maybe whether MSA Pairformer hallucinates when there's a lack of coevolution. And we think that this is also just very important to check in protein language models. So yeah, the rest is just acknowledgements. This is our lab. They do great work, so you should follow the work that's coming out of our lab. We're pretty new, so keep an eye out for more work. And also thanking the Steinegger lab, which where they focused on sort of this MSA pairing problem. But yeah, no, I'm happy to take questions, and thanks for listening.
Awesome. Thanks, Yo. Let's thank the speaker. All right. So